Hello everyone, myself Dr. Poonam Duhan from Government College Jain, today presenting a lecture on antibody. What is antibody? Antibody are the part of humoral immune response that are involved in pathogen detection and neutralization. Actually, antibody or we can say antibody are protein. These are mainly proteins that produce from the B cell in bounded form, but further these work as independently, and antibody are mainly protein that help in clearance of antigen or foreign particle that enter into our body. Then antibody are produced by plasma cell, but once secreted can act independently against the extracellular pathogen and toxin. That means they also a important role in humoral cell immunity because they can independently work against the extracellular pathogen and toxin. Antibody bind with specific antigen on pathogen, and this binding can inhibit the pathogen infectivity by blocking key extracellular site such as receptor involved in the host cell entry. Antibody can also induce the innate immune response to destroy a pathogen by activating the phagocytosis such as the macrophages, neutrophil, which are attracted to the antibody bound cell. Then the history about antibody. In 1940, some facts are recognized about the antibody. These are generally bivalent, and antibody contain heptane that is heterogeneous with respect to affinity. Antibody are different class based upon their ultracentrifugation and on the basis of their electrophoresis. Now, about the structure of antibody. Antibody basically are the heterodimer, or we can say these are the dimer of dimer. Antibody made up of two type of chain, heavy chain and light chain. The heavy chain are linked with each other by the hydrogen or disulfide bonding. In the same way, sometime reason may be present or not. The two chain is heavy chain and two is the light chain. The molecular weight of heavy chain is about 50 kilodalton, and the molecular weight of light chain is about 25 kilodalton. So that's why the total antibody hydrodimer contain a approximate weight of about 150 kilodalton. Antibody attached glycan are critically important to the structure and the function of antibody. Antibody are heavy globular plasma protein or glycoprotein. This is the structure of immunoglobulin. Antibody also called immunoglobulin, or we represent in them by A and B. These contain to heavy chain to light chain, and in both heavy and light chain there is some variable amino acid present, variable portion present, that is called variable region of heavy and variable region of light chain. But some portion of heavy and light chain are constant because they contain the constant amino acid, that is called constant portion. Here the pink color showing the variable region of both heavy and light chain, and blue region showing the constant region. The heavy and light chain is joined to each other by the disulfide bonding. Immunoglobulin domain. Immunoglobulin is composed of two identical heavy polypeptide chain and two identical light chain that are bounded by interchain disulfide linkage. Each chain is composed of structural domain called immunoglobulin domain. These domains contain about 70 to 110 amino acids. The heavy chain in immunoglobulin may be of five types. This is the alpha, gamma, delta, epsilon, and mu. Each heavy chain has the two reasons. One is the constant and one is the variable, as we discuss in the back slide. Alpha and the gamma chain contain approximately 450 amino acids, whereas mu and epsilon chain contain approximate 550 amino acid. Then the light chain of immunoglobulin, these are of maybe of two type, kappa and lambda. All antibody have one of the two kind of light chain. They may be either of kappa or lambda, but never be kappa and lambda light chain together in an antibody structure. A light chain has two successive domains, one is constant and one is variable. The approximately length of light chain is 211 to 217 amino acid. These are the site of immunoglobulins. This is the light chain and it is heavy chain. This linked to each other by disulfide bridge, and there is a region called Hing region present between the heavy chain and that provide flexibility to the immunoglobulin structure, and the variable region of both heavy and light chain is called antigen binding site because they bind to a specific portion of antigen and play an important role in precipitation, reaction or clearance of antigen. This is the Hing region that join the heavy chains that present between heavy chain, but it always present by in the alpha, beta and gamma, but never in the mu and epsilon. So IgG and IgM carry this Hing portion. Hing reason क्या है? Hing reason flexible amino acid stretch is the central part of heavy chain of the immunoglobulin G and immunoglobulin A classes, which link these two chain by disulfide bond. High content of proline and hydrophobic residue flexibility assist initiation of the complement cascade. Rich in cysteine and proline amino acid, extremely variable in amino acid sequence. No resemblance to any other immunoglobulin region. Gamma, delta, and alpha chain have the Hing region. Mu and epsilon chain do not have Hing region. Then the function of carbohydrate. All type of immunoglobulin contain a carbohydrate portion that is located between domain CH2 of the two heavy chain. The function of carbohydrate is it increase the solubility of immunoglobulin and it provide protection against degradation. It enhance the functional activity of the Fc domain. Thank you very much for watching this video.